Hello everybody, just a little tutorial for me again, just to go through some of these key terms and what happens to animal cells and plant cells when they're put into different solutions, right? And what these key terms mean. So just start going through the top set here. So these animal cells, we put these into an isotonic solution. Now, if say what an isotonic solution is, we have the same or equal water potential both inside and outside the cell. Yeah, so the solute is the same concentration. So same water potential in and out. Okay, so we've got movement in, we've also got movement out, no net effect. This means the animal cell here is awesome, it's fine. It's in an isotonic solution. Solution is not losing water, not gaining water. As soon as we put it in a hypotonic solution, this means now that we have a higher water potential outside the cell. Okay, so there's a lower concentration of solute. Yeah. So, for example, you would put this cell maybe put in distilled water, so we have a much higher water potential. Water rushes in through the membrane. A little bit will come out, but the net movement is in. So the pressure on the inside of this membrane actually bursts the cell and damages the cell. So we call that lysis. Cell lysis. It splits. If it's a red blood cell, which I was trying to draw here, we call it hemolysis. All right. So third solution, hypertonic, means that we have put the cells in a solution, a strong solution of salts or sugar. Okay, and we have a higher water potential inside the cell, so therefore water moves out rapidly through the membrane. Some goes in, but not much. The net effect is that we have a shriveled cell. This is called crenation. Okay, we see a cell that's much smaller than the one in isotonic, but the membrane is the same sort of surface area, but it's just been shriveled because we've lost all the water in the cytoplasm. All right, stick with me. Plant cells are a bit different because we have the presence of the cell wall here, which is kind of like a porous cage, if you like. It holds the cell in place. So in an isotonic solution, the cell is, the membrane's pressed against here, but it's not with any pressure because we have same water potential pressure both inside and out. So we have water moving in, water moving out. The cells really are flaccid, so what I mean by that is there's not a great internal pressure, osmotic pressure happening on the inside of the cell. If this an animal cell, there's no cell wall. Obviously, we'll move on to that in a second. Let's do it now. We'll move on now. So this is a flaccid cell because it's in an isotonic solution. Now, the hypertonic solution, yeah, we have, maybe it's been put in distilled water. There's a lot of fresh water around the cell. We have a higher water potential outside the cell, lower water potential inside. So water does start rushing in to the cell again. So as if it's been put in distilled water, maybe water rushes in, some out, net effect is water moving in. So because of this cell wall, this kind of cage-like structure around the membrane is now pushing against the cell wall with a high amount of osmotic pressure. If it was an animal cell with no cell wall, it would explode, but it doesn't. What happens now is we have that pressure on the inside forcing against the cell wall, which gives it turgidity. So we call that once a plant's got lots of water in it, around its roots, it can take up the water. Its cells then become turgid, and it helps it stand vertically up as it would like to catch the light. That's an adaptation of plants. They need to do it vertically to catch the light. To strengthen the stem, to support its weight, it needs that turgidity. Plant that has no water or not been watered for a while becomes flaccid. The cells become flaccid and the stem starts to fall over, and you see the leaves start to shrivel up. Extreme conditions, hypertonic solution now, which means we put this in a very strong salt or sucrose solution. Water rushes out. I haven't drawn the cell wall here. Oh yeah, I have, sorry. I have drawn the cell wall. Water rushes out, some in, but the net effect, again, is out because we've got a high water potential inside and a low water potential outside. So what we can see here, this structure here is the cell membrane. And you can see actually it's lost so much water that it's peeled away from the cell wall, which is there, and shrunk and shriveled, almost like this crenation effect here, but it's in a plant cell, and we call this process not crenation, plasmolysis, and we can see that under the microscope in our lesson this week. Okay, so all those key terms there. Move my picture, and they're all labelled here, and we've hopefully explained that for you. Cheers, guys. Thank you.